Okay, I think everyone has had a chance to join us, so we are going to get started. Greetings, everyone. My name is Kathleen Carter. I'm a bioinformatics data scientist here at RINC, and it's my pleasure to welcome you today to the National Consortium for Data Science webinar. I'm especially excited about today's session because our speaker, Dr. Karamarie Fecho, will introduce you to a tool that fundamentally changes how we approach biomedical research, helping us uncover connections we might never have imagined, and doing so with speed. Kara is the founder and CEO of Copperline Professional Solutions, a biomedical consulting and communications company she established in 2010. Copperline supports a variety of academic, industry, and nonprofit organizations and specializes in providing expertise in clinical and biomedical knowledge, discovery, and in translating scientific insights into actionable outcomes and tangible products. Kara's connection to RINC and UNC runs deep. She's been engaged with RINC as a research affiliate on numerous large-scale collaborative open-source projects for over a decade. Of note, she's been instrumental in developing ROBOKOP, which stands for Reasoning Over Biomedical Objects Linked in Knowledge-Oriented Pathways. I have a privilege of being part of the ROBOKOP team, and I can tell you firsthand that this tool is truly transformative. What if you could ask a computer, what connects this gene, this drug, and this disease, and get back biologically plausible answers grounded in real data? ROBOKOP is a biomedical knowledge graph system that lets you do just that. Traditionally, you'd spend weeks or months searching through literature, connecting the dots manually. ROBOKOP integrates roughly 10 million biomedical entities, 130 million relationships from over 40 curated data sources into a knowledge graph that you can query to test mechanistic hypotheses and propose new ones. What I think you'll find most valuable today is Kara won't just tell you about ROBOKOP, she will show you how it works through a live demonstration. Whether you work in academia, industry, or government, if you work with biomedical data, you'll leave this webinar with a new tool in your arsenal to accelerate scientific discovery. Please join me in welcoming Dr. Karamarie Fecho.
Thank you, Kathleen, for that very kind introduction. Let me share my screen. Okay, is everyone seeing my screen? Yes? Okay, good.
Good, looks good, Kara.
Okay, so as Kathleen mentioned, I'll be discussing ROBOKOP today, which stands for, as Kathleen said, Reasoning Over Biomedical Objects Linked in Knowledge-Oriented Pathways. So as such, I thought I'd just start with sort of an overview of what is this thing called knowledge. A lot of you were probably familiar with this DIKY, or data to information to knowledge to wisdom pyramid. But at the bottom layer, it all begins with the foundational layer of data, which is just discrete observations or facts that somebody made in a lab, on the field, or whatever. But they don't have an inherent meaning in and of themselves other than they exist. Information is sets of these data points that are somehow related to each other. So it adds a little bit of context to data and provides a little bit more meaning. Knowledge is curated information or things that are generally believed to be true. And that's sort of the layer with which ROBOKOP sits at, that knowledge layer. Wisdom is where the action happens. So wisdom refers to sort of those moments of eureka or serendipity or insights that you weren't expecting, but that were derived from the underlying knowledge. And therefore provided a new hypotheses or something new to explore, which loops you back down to data again, and the need to collect new data to further support or refute whatever your hypothesis is. And so ROBOKOP sits at that knowledge graph layer. And hopefully I'll show you how we can derive wisdom from ROBOKOP. While the data to information to knowledge to wisdom pyramid is very valuable as a theoretical construct, and it's been in place for almost 100 years, in modern times, knowledge tends to be represented in a computable graph form as a knowledge graph. So the term itself was first introduced by Google in 2012, but the general concept's been around since the 60s or 70s. Knowledge graphs are ubiquitous. We use them every day, whether we know it or not. So they drive Google search engine, Wikimedia's Wikidata store, and have a whole other applications across sectors of society. Now, the basic unit of knowledge graph in a knowledge graph is something that's referred to as the semantic triple or the subject predicate object triple. This is a machine readable, computable form of information where you can express things in a manner where it can be interconnected. So you get a whole bunch of these triples interconnected, and there you have a knowledge graph. In the example shown on the right, I highlight one single triple, which states that Da Vinci painted Mona Lisa. Knowledge graphs build on controlled vocabularies, ontologies, and data models. So a controlled vocabulary is simply like an organized arrangement of preferred terms, primarily used to index or retrieve content within a specific domain. So MESH is an example we're all familiar with, probably medical subject headings. So this drives PubMed, for instance. Ontologies are controlled vocabularies, but they also add relationships between terms. So it's not just the terms themselves, but it does describe relationships between them. An example might be Mondo or monarch disease ontology. BioLink model is very specific to the biomedical space, but it is an hierarchical graph-based data model and an upper-level ontology. So it sits on top of these control vocabularies and ontologies to provide some harmonization across them at a very high level. And so and on the right is shown some brief branches of the hierarchical trees. On the upper side, it's for the node or categories, and in the bottom, it's for predicates. And as an hierarchical data model, you can see that it starts off very generic. So the root of the category or node tree is named thing, and then it gets more specific as you go down the tree. So biological entity, disease or phenotypic feature, and then disease. And the same is true for the predicate tree. Starts with the root of a related to and gets more specific as you get down the hierarchy. At the core of BioLink model is this concept of an association shown as an onion diagram here. So on the left, you can see the basic structure of this association. At the heart of it is your core triple, so your subject, predicate, object. But each of those, or some of them, can be qualified to allow more nuanced assertions. And that forms a statement which itself can be qualified. On the hypothetical example shown on the right, the example here would be glyphosate is correlated with cancer. And this is just a hypothetical example, but glyphosate is the core ingredient in Roundup. So some of you may have seen the news recently related to Roundup, which is why I picked it here. But you can qualify that. So you have your core triple, which is glyphosate is correlated with cancer, but then you can say glyphosate in agricultural products. So I mentioned Roundup is correlated with, but maybe with long-term exposure and cancer, but specifically cancer of the prostate. And so this is how BioLink model allows you to make some really nuanced assertions in a computable form in a graph fashion. But what's in a name? So I presented that core triple in that hypothetical example as glyphosate correlated with cancer. But biomedical entities often are referred to by different names, different identifiers based on different identifier systems. So there's this whole big mess of having all these different representations of what is exactly the same thing. So this presents a bit of a problem. And with BioLink model and the NCATS translator program, we found a way to kind of overcome the challenge programmatically. So we developed two tools, Name Resolver and Node Normalizer, to sort of provide entity resolution to account for that mess in name and identifier disparities. And so Name Resolver takes a lexical string and it maps it to identifiers from a vocabulary ontology, as well as to BioLink model categories. So shown in the example here, the input is clofibrate. It's a different chemical. And then you can see it maps to the preferred identifier in BioLink, which is Kebi 3750, a bunch of cinnamons, and then some BioLink category types. Node normalizer is complementary. And in contrast, it accepts an identifier. In this case, it was Kebi 3750. And it returns the BioLink preferred identifier, all equivalent identifiers, as well as the BioLink semantic types. And so Kebi, in this case, is the preferred identifier for clofibrate. So these tools are really helpful. Between BioLink model and tools like Name Resolver and Node Normalizer, we developed a RoboCop architecture shown here at a very high level, but with like 4 key components that allow us to perform ontology-supported queries and have a harmonized system of interoperable graphs. So in addition to BioLink model, which we leverage for semantic harmonization, we have the 2 tools, Name Normalizer and Node Normalizer, which serve to provide entity and identifier normalization. But we also have a custom software pipeline called Orion, or operational routine, for the ingest and output of networks. And what Orion does is it ingests RoboCop's underlying knowledge sources, so the primary knowledge sources, and harmonizes them and normalizes them using BioLink model and tools like Node Normalizer. And then it integrates them and it outputs them as a collection of semantically harmonized interoperable graphs, including the RoboCop knowledge graph. RoboCop's architecture also has a governance component to it, so not technical, but socio, if you will, and that's DMOG, or the Data Management Oversight Group. And this group is responsible for evaluating new knowledge sources for compatibility with Orion and in ease of integration and ingest, and then prioritizing those knowledge sources for ingest into Orion and incorporation into RoboCop KG. So with this architecture in place, we now have this RoboCop knowledge graph system, which is not just one component, but it's a collection of modular interoperable components that serve different purposes for different users and different needs. So we have the RoboCop user interface, or UI. Shown on the right is the main landing page of the RoboCop UI, which can be accessed at robocop.renc.org. We have the RoboCop knowledge graph. We have a bunch of different API endpoints for querying the RoboCop knowledge graph. And then the Orion, or RoboCop collection of standardized and interoperable KGs. So not just the RoboCop knowledge graph, but also many of its underlying sources and some sources that aren't integrated into RoboCop KG. And then there's supporting tools and functionalities, such as an enrichment algorithm. So the RoboCop knowledge graph currently contains about 10 million nodes and 130 million edges, drawn from about 40 or so biomedical data sources and ontologies. Examples are comparative toxicogenomics database, or CTD, drug bank, binding database, monarch disease ontology, or Mondo, gene ontology, or GO, and human phenotype ontology, or HPO. A high-level schema is shown on the right over here. So the RoboCop knowledge graph can be queried and accessed by going from the main landing page to a RoboCop graphs page. And I'll show you that when I do the demo in a minute. But here you find the RoboCop KG and many of its underlying knowledge sources. So this is just a snippet from that overall page that I'm showing you here. And you can do a lot of things from the RoboCop graphs page. So you can go to the graph website link and then get access to a Neo4j endpoint where you can submit a cipher query. You can go to this details box here and have a whole bunch of different things you can do, including a download, a bunch of different API endpoints. There's a Sankey diagram, which helps you sort of visualize RoboCop's knowledge sources and how they interact. And then there's live updates on number of nodes and edges that are contained in the RoboCop KG as well as other KGs. You can also query RoboCop by going to the question builder as part of the user interface. So here on the bottom right, you see a welcome to RoboCop snippet. This links out from the main landing page. And here users can view example queries, the things that have already been preloaded, explore templates, so having things structured where you input whatever the terms are that you're interested in, and then RoboCop works as a template in that case. Or you can create your own query. And that's shown here in the question builder. So you can either use the fields on the left to do that, or you can use the bubble graph on the right to build your own query. You can also upload your own query in JSON format. And then after you submit it, it goes to the RoboCop Answer Explorer, which I'll show you in a few minutes. Okay, so that's sort of in a nutshell what RoboCop is, knowledge graphs and stuff like that. And I thought I'd provide one example application of RoboCop from a recent collaboration with investigators at the UDN, or Undiagnosed Disease Network. So the UDN is a prospective ongoing study of participants who have some sort of inexplicable disease at study enrollment, but with no known diagnosis. And many of these, once they're enrolled, they undergo really extensive clinical diagnostics and phenotyping, blood work, all sorts of things, omics testing, everything like that. And many of them end up with a diagnosis and effective clinical care, but a significant portion don't. And so the UDN investigators came to us because in the absence of a diagnosis, it kind of begs the question of maybe other components such as environmental determinants of disease. Um, and so the example that I'm going to show here is for participant UDN 235962, which is a male pediatric participant. Again, no diagnosis, maybe environmental determinants, presenting with a phenotypic profile of roughly 30 HPO or human phenotype ontology terms. There's a suspected gene, this EZH2 gene, which UDN investigators thought may or may not either contribute to disease in the phenotypic profile they were observing, or maybe causes disease, but they weren't quite sure. And then importantly, this participant had environmental exposure survey data available. So anything we could identify using ROBOKOP or hypothesize might be related to this participant, we could then go back to the survey data to check. Okay, so what we did first is we just asked a really, really simple question and asked, what BioLink chemical entity is BioLink related to that suspected gene, EZH2? And shown on the right is a snippet of ROBOKOP's results. And they're scored and they are ranked. And you can see that while we asked for chemical entities, some of these results that were returned were actually more specific than that. And that's through ROBOKOP's ontology-supported query and answer exploration capabilities, supported by BioLink model and some of these tools that we're leveraging. So in the example here, we asked for chemical entity, but we got back tretinoin, which is a drug, or Kebi 15367. We also got back a small molecule, or CPI 1205 with a uni identifier. And then of interest to our team, one of the top 12 results was bisphenol A, which is an environmental hazard that's produced during the production of polycarbonate plastics. So potentially interesting and potentially relevant to this particular participant. So we dove into that particular answer, the one for bisphenol A, and shown on the upper left is the answer subgraph for bisphenol A. And you can see that ROBOKOP returned 4 edges. One of those edges was an effects predicate. So bisphenol A affects EZH2. But when we looked at the provenance and evidence for it, we actually see that it was qualified. So it was more specific. It actually says it increases molecular interaction with. So the qualified triple in this case is not just effects, but it's bisphenol A increases the molecular interaction with EZH2, the suspected gene of interest. And this edge was provided by CTD, and it came with publication support in the form of a PMID. You also notice that one of these edges and occurs together in literature with edge, which we did not ask for as part of the query, but was added by a ROBOKOP service termed OmniCore to the results when it's available. And OmniCore exposes counts of co-occurrences of terms, in this case EZH2 and bisphenol A, in PubMed abstracts. And so this OmniCore service provides additional evidence in support of an association. In this case, there was 14 PubMed abstract co-occurrences of bisphenol A and EZH2. And then OmniCore is also used in scoring and ranking of edges in combination with some of the publication support provided by curated knowledge sources. So this patient had over 30 phenotypes in its overall phenotypic profile. So we decided to perform ROBOKOP's enrichment functionality and then provide a subsequent filtering step based on that gene of interest EZH2. So we input all of these different HP terms that were provided by the UDN investigators into the enrichment feature. And then we did a subsequent filtering step. And the first one that came back was for Weaver syndrome, which is a genetic disorder. And the predicate was causes, and EZH2, mutations in EZH2 are the primary cause of this syndrome. And it's characterized by accelerated growth, distinctive facial features, and some intellectual disabilities. And so we then thought that maybe Weaver syndrome is a potential diagnosis for this participant. So again, a hypothesis was put forward. And then finally, we asked what adverse outcome pathway might link bisphenol A to Weaver syndrome. So again, Weaver syndrome may be something that we want to look further into. And so we structured it very, very simple, AOP, using just the root predicate related to for the edges here. And then we looked for genes, biological processes, and phenotypic features that may link bisphenol A to Weaver syndrome. And shown on the bottom left is one of the top answer subgraphs, a portion of which reads in natural language that bisphenol A decreases the expression of GABA receptor A1, which is actively involved in GABA signaling pathway, which is negatively correlated with bilateral clonic-tonic seizure, which has a phenotype of Weaver syndrome. And of relevance, the participant's phenotypic profile did include a variety of seizures. So what this led to is action. So the knowledge from ROBOKOP then generated new insights and actionable outcomes. Specifically, ROBOKOP put forward a couple of hypotheses that one, perhaps the UDN participant or the participant's biological mother had a history of exposure to bisphenol A. Perhaps the UDN participant actually has Weaver syndrome. Maybe that was overlooked somehow in the clinical diagnostics. And so we put we now have next steps that we can take. We can go back to the environmental exposures data as well as the clinical assessment records and see if we can find any evidence for bisphenol A exposure. Again, this participant had blood samples available. They also have that survey data, and bisphenol A was one of the questions in the survey. We can put forward a hypothesis to the UDN investigators that they might want to consider a diagnosis of Weaver syndrome. And then I highlighted bisphenol A in this example here as a chemical of potential interest, but ROBOKOP actually generated a whole bunch of different chemicals that seemed of interest and that we're looking further into now to explore. Okay, and with that, I will acknowledge my team members and especially our funders. So ROBOKOP is currently funded by NIEHS and the NIH Office of Data Science Strategy. The prototype was funded by NCATS. So thank you for your time. I have my contact information listed here. You will be getting a copy of the slides sometime after this webinar. Please feel free to reach out to me or just go straight to the ROBOKOP team at robokop.renci.org/contact if you have any questions or want further information on ROBOKOP. And up next, I will stop sharing and we can switch to a demo. Okay. And I see there's questions in the Q&A, but I think we're going to go through those at the end of the demo. I'll be sure to leave plenty of room time. Okay. Let's see how this goes. Okay, are you all seeing somebody give me a thumbs up if you're seeing my screen.
Looks good, Kara.
Okay, thanks. Okay, so I had a screenshot of this in the slides that I showed you, but this is the actual robokoprenci.org main landing page. And I'm going to take you through a few different features just to give you a feel for how this works so that after this webinar, you're more prepared to kind of just play around with the tool itself. So I'll start here with the Explore the Graphs page. And if I go here, I had a screenshot of this in the presentation, but this is like the live page. And you'll see a tile for the ROBOKOP knowledge graph, but you'll also see a bunch of other tiles. These are for some of ROBOKOP's underlying knowledge sources, as well as some additional knowledge sources that have not been integrated into ROBOKOP KG at the moment. And from here, you can see the information on the nodes and edges that are available in ROBOKOP. If you go here, you can go to a Neo4 browser. Yeah, you do not have to log in. You can just submit your Cypher query here. And then let's see. If you go to details, this is really handy to explore what's available in RoboCop. There's a lot here on the detail page, so you can see prior releases of RoboCop with the versioning history available there. You can go back to that Neo4j endpoint, which I'm not going to go to here, but you can also go to an Automate endpoint and submit a variety of different types of queries. This one's usually handy. It's a knowledge graph metadata endpoint for RoboCop, so you can execute it. And then you can see a whole bunch of metadata that you can download in JSON format for further exploration. What else can we do here? But you can also go here and see a Sankey graph. And this is kind of cool because it shows you by primary knowledge source what predicates are exposed. And so it just gives you a visual feel for the relationship between primary knowledge sources and the types of predicates in RoboCop. And this is not only just generally informative, but it can help guide your queries a little bit. Additionally, if you scroll down, there's predicate counts. So there's multiple ways of sort of viewing the predicates. There's counts of predicates over here. And if you scroll down further, there's the node prefixes or query prefixes. These are just the identifier prefixes that are supported by RoboCop. Again, this is helpful in formulating queries. Edge properties that are supported by RoboCop, the primary knowledge sources and the number of edges contributed by each, and then aggregated knowledge sources, which are knowledge sources that are aggregators. So they ingest primary knowledge sources, and we pull some information from several aggregator knowledge sources, such as Drug Central, for instance. And so then there's just a whole bunch of other stuff like node properties. So the details page is very detailed, but it provides a whole lot of information that can help you explore RoboCop, the knowledge graph, and also better formulate queries. Let's see. So let's now go back here. So in addition to exploring the RoboCop knowledge graph and all of its underlying knowledge graphs, primary knowledge graphs, you can also go here. And this is what I was showing you with the UI example. So this leads you to a Welcome to RoboCop page. Here you can view examples. So these are preloaded things, and I'll try this one with Castleman disease. So this is asking about chemicals that might interact with Castleman disease, which is a funky, rare immune disorder that essentially, if untreated, causes multi-organ failure. So this is already pre-structured for us, and you can see in this case, we are asking for a gene that's somehow related to Castleman, but for a chemical that specifically interacts with any of those genes that are returned. And so this would be sort of like a drug repurposing type of question where you're exploring chemicals that might do something to a gene that is causal to Castleman. And if you submit it, it takes you to the Answer Explorer page. And when looking just through these answers, I can tell that the first two are good answers, if you will. The first chemical that's exposed here is widely used off-label for the treatment of a subtype of Castleman disease. And the gene that's shown here plays a strong role, IL-6 receptor signaling in Castleman's disease. And so you can sort of play around with it, move things around, so it's easier to look at. You'll see a lot of these occurs together in literature with edges. We are sometime soon providing a toggle, if you will, or an option to sort of allow you to turn those off, because as I'll show you in a minute, sometimes there's an awful lot of these edges and it gets kind of hard to read. They're really informative, but sometimes you just want to look at it without them. And then I can tell that the second drug that was returned here, or a chemical, but it's a drug, is actually FDA approved for the treatment of a subtype of Castleman. So that's just demoing an example query. We can also export the templates. And there's a number of templates here, and I'm going to do the AOP since I showed you an AOP in this slide deck. And we're going to do particulate matter asthma. So a different one than I showed you, but still an AOP. And let's see, select the query. And then it's already structured, so it's just a generic template like the one I showed you in the slide presentation. And then here's your answer, your result set. And you can slide this around a little bit so it resembles your query. Let's see, this was like this and like this. Okay, so there's the basic structure of the AOP that was asked. And we can look at the first one. And the first answer has TNF as a gene, inflammatory response as a biological process, and cough as a phenotype. So this seems very reasonable for something related to asthma. And when you look to see the answer graph that you can explore, this is why I was saying we're going to be including some sort of toggle to get rid of some of these co-occurrence edges or the omnicore edges, because it makes it kind of hard to read, but you can sort of see, let's see if we get it structured a little bit, the general structure here. And you can see there's a whole bunch of different edges that some of them are really specific. So asthma is manifested, or cough is a manifestation of asthma, I should say. Let's see, TNF is positively involved with, so clicking on that edge brings up the provenance with inflammatory response, and it gives you some of the information here. This one didn't have any supporting literature, but it was contributed by HedionNet as the primary knowledge source. So that's shown in the box there. Let's see. And again, we have these co-occurrence edges. So particulate matter co-occurs with cough in 153 PubMed abstracts. And you can just keep exploring as you will. And glancing at the overall result set, this looks very relevant to the question that was asked in terms of the types of genes that were returned, the phenotypes, and the biological processes. So inflammatory response are pretty dominant here, but I think further down you get a little bit more specific biological processes. You can increase the page size. So if we go to 100, that expands the view a little bit. So you can scroll down a little bit more quickly to take a look at your answer sets. Let's see. We can go back to the question builder. Actually, let's go back to this page and then over here. And now let's create a query. And I mentioned that glyphosate and Roundup are in the news a lot lately. I don't know if anyone you saw, but a few days ago, The Guardian published an article that landed in my inbox stating that a paper in Regulatory Toxicology and Pharmacology that was published in 2000 was retracted. The paper focused on the safety of glyphosate, so things are now being reconsidered in terms of safety. And Monsanto, the company who makes Roundup, has a number of lawsuits from people who are either in the agricultural or landscaping industry, things like that, and various bad outcomes. So this is in the news. It was timely. I thought maybe we'll show you this as just an example. And I also want to highlight something that I didn't provide in this slide. So this is just a real quick and dirty query. I'm not choosing a specific predicate, just leaving it at the root of related to. But I'm going to ask for named thing, which is the root of the BioLink category tree. And so this is just tell me everything you know about glyphosate, essentially. And we can submit this, and there's a whole bunch of things that are returned. We can scroll down a bit. Maybe expand that view to, let's see, 100. And we ask for named thing, but we're seeing mostly like phenotypes and diseases here. There's an enzyme that's returned there that might be a target. And it's probably not, I don't know much about glyphosate, but it's probably not terribly surprising since the target is a plant enzyme, and I think it acts somewhat indiscriminately in humans. But this gives us something to explore. We see agricultural worker diseases, some prostate disorder, some cancers, things of that. So just something worth to point out and to highlight that named thing functionality. Now, in this example, we didn't see too much variety in the types of BioLink categories that were returned. There was an enzyme, but it was mostly phenotypes and diseases. But if we change that to something else like albuterol, which is a drug that is used to treat asthma, you can submit that. This might take a second because there's a lot. This is an old drug. Okay, so we get a whole bunch of answers, and if we take a closer look at them, let's expand the view. I'll do 50. We can see diseases as well as genes and proteins. And I maybe should point out, so this one here is for the adrenergic receptor beta 2, which albuterol is an agonist for. So that's good that it was in the top answers. But genes and proteins are conflated in RoboCop, so that's somewhat important to keep in mind when you're using it. And that's primarily because they're conflated in our underlying knowledge sources. So people refer to both genes and gene products kind of interchangeably. So RoboCop conflates them a bit. Let's see. So those are some of the options for exploring the graph and asking a question. There's a few other things that are available up here on the header. One of them is going back to that graphs page. So you can get to that graphs page from Explore Graphs or from Explore the Graphs, that big purple tile. But there's also a node search operation here. So you can put in, say, you can search by identifier or by name and show you what is returned. There's pretty much a lot of stuff related to asthma. So this lets you explore synonyms, more details on these different compounds. So we can go over here. It tells us a little bit about the definition of at least what Wanda thinks asthma is and a whole bunch of other things. Let's see. You can also get to the enrichment analysis. Maybe I'll show one other thing, but so you can get to an enrichment analysis. I actually have this queued up over here. Here in the enrichment analysis, it performs an enrichment. So in the example I showed you, I input a set of phenotypes, and those are the ones I input into the enrichment function. To perform the enrichment analysis with diseases. So it's a many type of operation. So it looks for how closely one or more of these phenotypes are related to, in this case, a disease of interest. And it basically looks at their relatedness and connectedness based upon some intermediary phenotypes. Okay, and we can change that to disease. These are all the BioLink classes. Submit the query. Okay, and so now you can see the results of the enrichment. Now, in the example I showed you from the UDN participant, we then took these results and applied a filtering step by that gene of interest, but this is just the raw enrichment results. And you can see they're supposed to have p-values, but I'm not seeing that here. But you can download these results as a CSV file. Let's see. Oh, this is also available. So what these are predictions on drugs that may treat diseases. So these are drug disease pairs that were generated using graph embedding and subsequent neural network modeling. And these are also ranked and scored. These do have actual scores on them. And you can do a bunch of stuff here too. This is sort of a new feature that allows you to just explore things for further research, perhaps for drug repurposing or something like that. But oops, not filter. So if you filter it, should just show you, yeah. So this is narrowing it down to just diabetes in this example. And let's see. What else? Developer tools. Okay, so we're going to add something new to this developer tools, but this is the RoboCop KG, so it takes you to that Neo4j browser that you can get to also from that graphs page. There's a number of different ways to get to some of these same functionalities, and that's being really slow. And that's where you can submit this is the same, so this is where you'd submit your query. And these are the phenotypes that I showed you in the enrichment and also in the slides for that UDN participant. So this should work to give you so this is just the results of a Neo4j query. And keep in mind too, like querying the RoboCop knowledge graph is by Neo4j is not going to return quite as rich of results as you would get through the UI, just to keep that in mind. But you can run the query. And then this takes you back to that Automat API page where you can run a bunch of different operations. I showed you the metadata endpoint. And we're going to add something here that's Kathleen noticed was missing, which is a link to the GitHub repo, which is available here, but it's kind of hidden a little bit. So all of RoboCop's sources, it's open. It's all publicly available, and we'll have a new tile here soon to link directly to the repo. And let's see. So very importantly, let's try that. This has a number of things, including just a description of what RoboCop is, licensing information, funding information, some citations. Where did the funding information go? But citations, events is in there, but there's no upcoming events posted. And importantly, there's a support guide, a tutorial, and contact page. So this is a good way if you find a bug or something like that, you can let us know from here, or once we have that tile in for the GitHub repo, you can just go straight to the tile to post a ticket or create a new issue. And I think I've pretty much gone through most of the key things. And Kathleen, unless you see something I'm missing, maybe we can open it up for questions.
No, I think you did great, Kara. Thank you so much for this. It was a
Oh,
wonderful talk
absolutely.
and demonstration.
Oh, there was one other thing I didn't say.
Sure.
There's an optional login. You don't have to log in though, and I'm not logged in apparently here, but just so you know, in the future that'll allow you to create your own personalized workspace. So,
And we have had an abundance of questions for you. I am trying
oh,
to stay on top of some of them so that hopefully we'll get to everyone before the end of this. So maybe we can start with some of them that we haven't touched on and then in the chat, and then we can talk about some of the ones that I've answered as well. And again, anyone that does not get their question answered or we don't have time to get to today, like Kara has said, please reach out to the RoboCop team. We are happy to work with you and really enjoy working with others in our knowledge graph. So we always encourage that open collaborative environment. All right, so we're going to get started with a couple of questions that we received prior to the webinar,
okay.
and then we will move into the ones in the chat. So the first one, Kara, is how can this package be integrated into a curricular program?
Okay, so that's a good question. So RoboCop has been integrated into a computational biology course at the NC School of Science and Math. So this is like juniors and seniors in high school. That's the only curriculum for which it's been integrated into, to my knowledge. But I could see it there's an environmental health or toxicology component to RoboCop. It's kind of skewed a little bit, in part because a lot of our users are toxicologists are from NIEHS, our funder, or from EPA, and this area is just kind of rich in that. So I could see it getting integrated into, say, a toxicology course. It's used just as a supplementary material, right? So it like Wikipedia, like ChatGPT, like anything else. That's my understanding of how it's being used right now. Just another piece of your academic toolkit to help guide your learning.
Great, thanks, Kara. And I think if anyone was interested, we would be more than happy to help you add this to your curriculum if you wanted to set that up. Uh,
Yep.
okay, another question for you. How can RoboCop facilitate interoperable knowledge graph completion, i.e., inferring new triples, or help develop new methods to achieve it?
Okay, so RoboCop does not expose inferred or predicted edges. I did show you that drug disease thing. Those are predictions, but it's not within the RoboCop knowledge graph. I also showed you enrichment, which, it's sort of like gets towards the heart of that a little bit in terms of inferencing. But that's also a separate functionality. But RoboCop itself, like the KG, could serve as for benchmarking purposes for the development of new edge inferences and predictions and knowledge graph completion. And everything is downloadable. So it's all open source. So I think that's a good way to facilitate that. Kathleen, did you have anything else to add?
No, I think that's right. You got it. So yeah, we love seeing it used as benchmark. Yeah.
And
Next,
if you are, let us know. We
yeah,
would be happy to know that.
yeah, we would love to see your work and see what you came up with.
Yep.
All right, so next question. How does RoboCop tell truly relevant paths from ones that are just highly connected but not meaningful?
So RoboKop doesn't. That is sort of up to the user to consider. And there's a good reason for that. So for instance, there may be users who really only want to see things that have a lot of evidence and is really highly connected. And that's like their comfort zone or whatever, that they just want to see absolute validated ground truth. And then others may be looking for, say, novelty and something that has hardly any connections at all, but there's one and it kind of looks interesting. And so different users kind of approach this differently. RoboCop doesn't just, it is what it is. But it does have all of its evidence and provenance and supporting publications and things that can help guide the user in confidence, or whether or not they want to further pursue any sort of observation based on edges and things.
Yeah, I think that's a really good point, Kara, that RoboCop is what it is. We just want to present the knowledge in the way that you can use it for whatever situation you are looking for. So we're not trying to impart our own biases on the graph that would, say, oh, we think this is more important or this is more relevant to a question because we don't know what our user's relevance or importance really is.
Hmm. Yeah, and just adding, like you'll see that this is a modular system. That's one of the reasons, right? Why? So some users may get a kick out of Cypher queries, and that's what they want to explore. I don't think I'm one of them, but like and then some people just want to use the UI.
Mhm.
There's different ways for people to download things. You can take those different pieces. So all of those different, like what I showed you on the graphs page, you can download each one of those RoboCop primary knowledge sources. Not all of them are on that page, but a lot of them are, for your own whatever use. So I mentioned benchmarking, that's one application, but it could be something else entirely. Oh, we should probably mention in the queue is this idea of creating your own personalized knowledge graph. So the UI will have another page. I don't know where, but like where a user can say sort of like, okay, I only want environmentally relevant knowledge sources in my personalized knowledge graph. And so then they can go and just pick and choose what they want. Maybe that's CTD and I don't know, but that functionality will be, it's in queue for this coming sometime in 2026. But I think a lot of users will find that happy. We've got many requests for that sort of functionality. So the flexibility, in other words, is intentional.
Yeah, yeah. I am going to combine a little bit two of these questions because I think they touch on the same thing, which is how can I use this and where do I get started? The questions themselves are, how do we implement knowledge graphs in our own Python programs? What is the best approach to get started? And is the graph accessible to AI or APIs? And is the advanced biomedical data included? Um,
And
yeah.
what was the last part?
Is advanced biomechanical data included? Excuse me.
Oh, okay. So advanced biomechanical data probably is not well represented, if at all, in RoboCop, but other types of biomedical data are. Um, and there's a lot of, I think these are all related. And I think I kind of touched on this previously when we were talking about flexibility and how you can download everything for free. You can go to the Explore the Graphs page, API calls. You can take this and use it for your LLM model in addition to benchmarking. It's basically, there's a number of ways you can use it for pretty much, and if you want to test your machine learning algorithms, you can use it for that. If you want to develop a new predictive model, you can use it for that. I don't know, Kathleen, any other
Yeah, and
ideas?
and for those developers out there that are looking to put this in Python, all of our source code is open access on GitHub. You are more
Mhm.
than welcome to fork our repo and get started that way. We've also made Orion, which is our parsing and like our ETL pipeline package available via pip install, so it'll readily integrate into your own Python environment. So if you have any questions about that, the GitHub repo is a great place to reach out to the developer.
Yeah, and it's very well organized. And like I said, Kathleen pointed out that we really need a tile on that one developer tools page. So that I think will be added like now, pretty soon. That should be easy. So
I think
that'll help you get to it. Right now, it's
it's
a little bit hidden somewhere, but um, we could probably put it in the chat too. So
yeah, we'll definitely share that link.
yeah, well, I'll add it to the slides so that it get distributed. That's probably the best way, so it's not disappearing after the end of this webinar.
Perfect. Thanks so much, Kara. And then let's see, let's go to some of our chat questions. Um, Here's one. Have people integrated knowledge graphs with causal inference approaches in observational studies?
Say that again. Have people integrated?
Knowledge graphs with causal inference approaches in observational studies.
Okay. This is a complicated question. So yes. I don't even know where to begin to answer this. So yes. So I don't know where to start. So for one thing, we do have a knowledge graph called, and it's available as an on that Automat API page and on the graphs page called ICES, or the Integrated Clinical and Environmental Exposure Service. We have used that graph. It's observational data. It's EHR data that have been integrated at the patient level with environmental exposures data. It's all regulatory approved and all that. So we don't expose any identifying information, but we've taken that data directly from the API endpoints and used it in multivariate analysis, including causal inference models. So it's readily available to that. Um, so that's sort of how we've applied that clinical observational data in the context of causal inference modeling. And that's published, so you can find the model itself, and the repo is open for that as well. Um, we also have not yet integrated, but one of the ROBOKOP knowledge graphs that you can access from that graphs page is called CAM, KP, which stands for Causal Activity Model. And in part of that too, it's kind of cool because we've basically taken these causal activity models derived from Go, or gene ontology, and supplemented them with some AOPs, or adverse outcome pathways, which are also causal models from the AOP wiki, if any of you are familiar with that, specifically the ones that have been endorsed, so that are generally accepted knowledge, not the ones that have not been endorsed, which are still in development mostly for the AOP wiki. So that's just another kind of approach that we've taken to get at causal things. But I think you can take any of our graphs and apply a causal model to it. And eventually the CAM data will be integrated into RoboCop. It just hasn't happened yet. We have had other priorities. So,
All right, so I've had a couple questions online about what is the score that is displayed in the answer question.
okay. I knew this was going to come up, and I didn't touch on it during the presentation, but the scores in RoboCop, so there's a complicated scoring and ranking algorithm. I did mention Omnicore. So that's the edges that are added by RoboCop to two entities as new edges in result set when available. Essentially, the RoboCop scoring and ranking algorithm has adopted or modified an approach from electrical resistance theory. So it looks to see how tightly or far apart things are in a graph in terms of their electrical resistance, and it bases some of that on the number of publications support. So weighting publications derived from curated knowledge sources like CTD almost always provides at least one publication, and that's CTD is highly curated. So that kind of knowledge gets ranked more heavily than Omnicore, which is just co-occurrences in PubMed abstracts. But those two pieces of information primarily, it also adds in p-values and things like that if those are available, but it's primarily based on those publications. Now, one important thing to note, so everything's scored and then ranked. And I don't know if you noticed, but some of the in the results set, some of the top answers, they're not going to be necessarily 1 or 0.9, which is the highest, and that's like the best, if you will. But these scores are all relative to a result set, and I wouldn't compare across result set because that won't apply. So you can't say a 0.6 here from this result set is better, or that answer is better than a 0.3 from a second result set. So that would be a misinterpretation. Just look at it relative to your result set.
That's great, Kara. Thank you so much. I know that's always a difficult question trying to explain that
It
score.
it is.
Maybe you could briefly touch upon how RoboCop and other related projects kind of touch on each other, such as the Proto-OKN and the translator ecosystem.
Okay, so that's an interesting question. In full disclosure, I am involved with the Proto-OKN, not through RENCI, but through a different project called BioBricks, OKG, or Open Knowledge Graph. RENCI is very much involved with the Proto-OKN as well. The funding sources for these are completely different. The goals are different, but there is some obvious synergies there and overlaps, but we do try to keep things very separated as much as we possibly can. But so, for instance, there's maybe tools like Node Normalizer is a very valuable tool. So that was developed in Translator, as I mentioned, primarily. But we adopted it for RoboCop because it's open source and we should be reusing it. So there's certain tools and things like that that just they do get reused in different projects, but the goals of those three different projects are very different. So for instance, the Proto-OKM project is funded by NSF. It stands for Open Knowledge Network, and it's got everything. So RoboCop is biomedical with a bent towards environmental health and toxicology. Proto-OKN does everything from supply and demand chains to like manufacturing and production and things like that. The biomedical piece in Proto-OKN is like really small. Um, and so these programs, there is a lot of overlap. That is true. Um, and so there's overlap in people. So I just said that I'm involved with Proto-OKN and actually Translator and RoboCop. And the same is true for some other people at RENCI as well and other institutions. But we do try to keep the funding and the goals of the different projects completely separated with the understanding that certain pieces of them that are really valuable, we're not going to rebuild and we're going to reuse. Kathleen, do you have anything to add to that? Because that's a good question.
No, I think that's really true that one of the things that's great about this project in Renzi is how important we think that our development should be interoperable and reusable as part of the FAIR ecosystem. And you're just demonstrated how is those things. And so is like the node normalizer, which is just another wonderful attribute of this project. We really want people to be able to reuse these things. So I think we have time for one more quick question. And
Mhm.
I'm going to say, can RoboCop answer more open-ended questions, such as the state-of-the-art research on involving cerebellum for cognition?
Okay. Open-ended questions. So I did show you that name thing. Cerebellum for cognition is like packed in terms of what it is there. So you might have to break it down a little bit, like into cerebellum linked to or cognition, and then somehow you can structure your question to sort of get at, I think, what you're asking, really by leveraging the name thing and related to. So you're not getting actually that's maybe a tip. I always start with very high level categories just to get a feel for what's available in RoboCop, and I might explore the metadata a little bit. And now that we have that details page, that's really helpful to see what's kind of predicates are in there and things like that, and what types of node prefixes are supported. But I think you can get at it through if you broke down cerebellum for cognition a little bit more and then added in a named thing depending upon how you might have to play around with it a little bit, but a couple of queries should get you there. And I'd probably start with cerebellum named thing, cognition named thing, and then piece it back together somehow. Do you have any tips?
Yeah, no, I think that's right. I think there's an art to developing your queries, and there's a little bit of puzzle pieces. But I know Amanda has a couple closing remarks, and we are at time,
Oh,
so I'm going to have to conclude this conversation. So I will kick it over.
thank you, everyone. I really appreciate it.
Yep.
And happy holidays.
Yes.
Thanks so much. And we are going to just add a couple of closing slides really quickly. I'm Amanda, the Associate Director for the National Consortium for Data Science. We are a part of RENCI, so we're grateful that you guys joined today. So grateful for Kara and Kathleen. Both of you guys did an amazing job walking us through all that content. Thank you so much. I do hope that folks will take a minute to join us at our next event, which happens to be a Careers in Data Science event. We are bringing in Becky Brown once again from SAS. To speak to how to use LinkedIn like a data scientist strategically. So hope you guys can all join that exciting conversation at the end of January. And other than that, please do take a minute to fill out our survey at the end of this webinar. We would love to know how we can improve events like these moving forward. Your feedback matters. Thanks so much, everyone. This was the last DataBytes event for 2026. It's been a pleasure learning